Hi, this is Bernard-Verdier. I'm calling from Berlin, Germany, and I'll be presenting today some work I've been doing on plant evolution in cities. More specifically, I'll be looking at phenotypic differentiation in response to both urbanization and neophyte invasions. So cities are hotspots of change, and these can be both abiotic and biotic changes. Typically, when we think of urbanization, we think of the modification to the physical and the chemical environment, which creates abiotic novelty. For instance, built areas creating impervious surfaces, or the urban heat island creating a microclimate. These are typically associated with biotic novelty, that is, the introduction of non-native species, creating novel biotic interactions with native species, and generally creating what has been called novel urban ecosystems. Altogether, these are expected to create novel and possibly quite strong selection pressures on organisms. I was interested in how plants are coping with this change. And first of all, many plants don't do it really well. There is a lot of local extinction and filtering out of certain plant species. A lot of natives don't make it into cities so well. And then for those native and non-native species who do make it in cities, there is generally a very high level of phenotypic variability, actually greater than in any other man-made system or natural system. These changes are plastic. A lot of very well documented plastic changes in plants have been documented in cities, and they can be evolutionary in nature, either via natural selection or genetic drift and isolation. Overall, we're still in the dark as to what are the main drivers of these phenotypic changes and what phenotypes are actually being selected. I'm going to investigate two questions. Is there evidence for heritable phenotypic differences along the urbanization gradient? And are these differences the same when we're looking at the abiotic or the biotic aspect of novelty in cities? I'm working in Berlin on dry grasslands. With my collaborators, we've been working for a few years now on a set of 56 plots of grasslands. You can see here on the map, and they are distributed along an urbanization gradient. For today, we're going to use a proxy, which is the percentage of impervious surfaces in 500-meter buffer around the grasslands. And this is typically really well correlated to, for instance, microclimate, temperature, precipitation, etc. And we're going to be interested in a second gradient, which is a gradient of biotic novelty, here captured by the proportion of neophytes in the grassland communities. The neophytes are recently arrived non-native plants, recently, that is in the last 5 centuries. Very often, these two gradients are quite positively correlated in cities. In our case, it's not so much the case. The correlation is pretty weak, which means we're going to try and see if we can disentangle the effect of both gradients on phenotypic changes of plants. This is what our plots look like. This is an old abandoned airport in the middle of Berlin. This is a very urban, almost rural grassland next to a road and an old railroad track. And these are, this is an example of a more almost natural grassland in the forest around Berlin. So we focused on 4 very widespread grassland species in Berlin: 2 grasses, Agrostis capillaris and Calamagrostis epigeios, and 2 Asteraceae species, Tanacetum vulgare, the tansy, and Solidago canadensis, which is a well-known neophyte and invasive species in Berlin. We used plant functional traits as phenotypic markers of change. Leaf traits, which allowed us to capture strategies of vegetative growth and resource acquisition. These 3 leaf traits are very commonly measured: specific leaf area, leaf dry matter content, and leaf area. Light interception and above ground competition was captured by vegetative height. And we used two traits related to reproduction strategies: reproductive heights, that is the height of flowers, and the onset of flowering. Our experimental design was in two phases. First phase was a field study where we sampled field populations of each of the species, measured traits on them, but also collected seeds. And these seeds were used in the second phase. They were grown in a greenhouse experiment in completely randomized, standardized conditions, and traits were once again measured on these plants. The whole idea is that we're going to be interested in comparing the traits of the mother populations in the field to the traits of the offspring grown in the greenhouse. And any differences that we find between populations in the greenhouse can only have been inherited from differences in the population in the field. How are we going to detect plastic versus heritable trends along the gradients? So the trick we're going to use is that we're going to think of any trends, any differences in the field populations as being a result of both heritable and plastic differences between populations. Whereas in the greenhouse, any trend, any difference that we find between populations is necessarily something that has been inherited. So we're going to use this, this basis to kind of detect whether we have heritable trends or not. Case number one, here I'm representing a graph which is going to be what I'm going to use for the rest of the results. The traits on the y-axis are varying along one of the two gradients. And here in this first case, the trend in the field was an increasing trend, a positive trend, but we found nothing in the greenhouse. That's the blue trend. This is a typical case where there's no evidence of heritability. Probably the trend in the field was only the result of plastic responses. Case number two, we found the same trend in the field in the greenhouse, and typically this is a very good support for the existence of a heritable trend in this trait. Third case is more of a mixed bag. In this case, we found a trend in the greenhouse, suggested that there is some inherited differences between populations, but this trend did not exist in the field. And this suggests that there is some more complex interaction with plasticity going on in the field. So going to the results, first along the urbanization gradient, I'm going to present all the results similarly in this sort of table with traits as rows and species as columns. Here for the first species, Agrostis capillaris, we found in the field only a trend for specific leaf area, which kind of tended to increase with urbanization. For the other species, we also found a few trends, not many, in the field. In particular, we found strong trends in leaf traits for tansy, Tenetsetum vulgare. And the question, of course, was, are they maintained in the greenhouse in the second generation? The answer was basically no. All four trends in the field were completely disappeared in the greenhouse. However, we did find some differences between population in the greenhouse. So this is our case number 3 that I presented before, where there are some heritable differences, but they're not necessarily detectable in the field. So to go through it progressively, we didn't find any inheritable trends in the greenhouse for SLA. We did find some for the 2 other leaf traits. For leaf dry matter content, it was variable depending on the species. For leaf area, we generally found a tendency towards a decrease with urbanization. We also found a decrease in vegetative heights, but nothing for reproductive height and nothing really clear for flowering either, although I could tell you about this a bit more if you're interested. So to summarize, with urbanizations, all trends in the field were actually lost in the greenhouse, suggesting that these were really mostly plastic responses. This was particularly the case for tansy, which showed really strong trends in leaf traits, but everything was gone in the second generation in the greenhouse. We did find some heritable trends in most species except tansy. And interestingly, we found general patterns of urban plants being less tall and having smaller leaves, which is kind of opposite to what is expected from the literature. And it's also opposite to what we see in terms of plastic responses in the field. This is interesting because a lot of the literature so far has been measuring plants in the field. So it is very likely that a number of things we know from field studies actually captures plastic responses and not really heritable trends. Second, the trends along the second gradient, the proportion of neophytes. So here again, we found quite a few significant trends in the field. In particular, for specific leaf area, we really had a general tendency towards a decrease with the increase in neophyte proportions. But once again, are these trends maintained in the greenhouse? The question is, the answer at this time is a bit more mixed. So generally not. We see that a lot of the trends disappear in the greenhouse, but this was not the case for one of the species. So specific leaf area for Agrostis capillaris, small, quite dominant grass in these grasslands, showed the same pattern in the field and in the greenhouse. So we really seem to have a heritable decrease in specific leaf area along this gradient. Leaf dry contents showed some weak trends as well, but otherwise vegetative height showed the strongest pattern, either a decrease or perhaps an increase depending on the species. For all of the traits, there was nothing really clear going on. So overall, once again, the field trends mostly disappear, except for this one species, Agrostis capillaris, who saw a heritable decrease in specific leaf area. And in this case, it seems like the inherited and plastic tendencies were going in the same direction. It's our only case so far. The very few heritable trends that we observed were not very consistent across species, and they also were in opposite directions to urbanization, which is interesting because oftentimes neophyte proportions and urbanization co-vary in cities. So here in this case, we see that they actually don't select for the same kind of traits, apparently. In summary, we did find evidence for heritable trait differences between populations. Whether these are based on genetic or epigenetic differences or maternal effects, it's hard to tell. But we do know that if they're heritable, then selection can happen. We found fewer trends with neophyte invasions, and when we did, they were often in opposite direction to the ones we found for urbanization. We found no heritable trends for traits which were related to reproduction. All the ones that we did find were for trends related to vegetative growth or above ground competition. We did observe very high levels of plastic responses. Some species were only responding plastically, like tansy. And plastic responses were often going in opposite directions to the heritable tendencies we observed in the greenhouse. This suggests that we should be quite careful interpreting field studies of plant trait variation along urbanization gradients, as these might actually not really reflect what's going on in terms of heritable trends. Finally, can we at this point talk about local adaptation? Not quite. We would need to go further and measure differences in plant fitness. However, we do find gradual trait variations along the gradients, and this means that it's consistent with gradual trait responses to the gradients. So, for instance, we could look at the shorter and smaller leaf plants in urban areas as an adaptation to drought, drought being one of the main constraints of urban living for plants. Patterns with the proportion of neophytes are a bit trickier to interpret. This could be a result of either a direct effect of the presence of neophytes or an indirect effect of the habitat selecting both neophytes and different kind of traits. This we can't answer so far, and we would need to, for instance, look at removal experiments or manipulation of the proportion of neophytes in these grasslands to go further. Finally, it's interesting to note that we found opposite selection pressures for the two gradients. And one may wonder if increase in neophytes in cities might actually be slowing down adaptation to urbanization. So I thank you very much for your attention, and we'll take any questions.